everyone, this is Teacher Lou from Delta Learning Space. Today we are going to be chatting about cell division and why cells divide. In this video, we'll learn what the cell cycle is, the purpose of the cell cycle, and the different stages in the cell cycle. Ready? Let's get started. But before we get started, I wanted to share a few resources linked in the description below. You'll find a free downloadable cell cycle vocabulary sheet and a graphic organizer. There's also an optional paid set of guided notes that mirrors this presentation and can help you follow along as we move through the material. The first question that we need to ask is, what is the cell cycle? The cell cycle is a sequence of events in which eukaryotic cells divide. This process allows for cells to grow, repair, and replace themselves. The cell cycle is divided into two main phases, interphase and mitosis, also called M-phase. In this video, we'll be primarily focusing on interphase. The version of the cell cycle that we're exploring applies to eukaryotic somatic cells. And somatic cells are also known as our body cells, and they make up most of the cells in multicellular organisms. Examples include skin cells, hair cells, and liver cells. In fact, the vast majority of cells in your body are somatic cells. Before we continue, it's important to note that cell division does occur in prokaryotic cells, but their process is different from what we'll be discussing here. The primary goal of the cell cycle is for one somatic cell to grow and divide into two identical daughter cells. Daughter cells are the cells produced from division, and they are genetically identical to the parent cell from which they come. And you may be wondering, so why does this matter? Somatic cells divide because this process allows organisms to grow, maintain, and repair their tissues. Organisms grow by increasing the number of cells through cell division. For example, humans begin as a single cell called a zygote. Through repeated cell division and differentiation, that one cell eventually develops into a complete organism. For this to happen correctly, each new cell must receive the same exact genetic information as the original cell. This image here shows a blastocyst, which is a cluster of cells formed shortly after fertilization. All of these cells are genetically identical. Here we can see how repeated cell division leads to growth. Our one zygote cell divided and formed two cells. Those two cells divided to form four, and those four cells divided to form 8 identical cells. An adult human is made of billions of cells, but every person begins as a single cell that must go through the cell cycle many, many times. Another important purpose of the cell cycle is repair. Cells can become damaged or worn out over time. Cell division allows the body to replace those damaged or lost cells. And not all cells divide at the same rate. Different types of cells divide more frequently depending on their function. For example, cells lining the human intestine are frequently damaged during digestion and divide about every 4 to 5 days. But in contrast, mature neuron cells do not divide at all. These specialized cells transmit information throughout the body. And because neurons do not divide, damage to the brain or spinal cord can have long-lasting or permanent effects. And here we have our nice little neuron cell in the corner. Now that we've explored why the cell cycle happens, let's take a look at what the cell cycle actually looks like. This diagram here shows a general overview, where the cell will go through G1, progress to S, go to G2, enter mitosis, and then repeat. As we mentioned earlier, the cell cycle is divided into two main parts, interphase and mitosis, sometimes known as M-phase. As you saw from the diagram, the cell will spend most of its time in interphase, where it grows and prepares for division. Those stages of interphase I've listed below as G1, S, and G2. Sometimes a cell may exit the cell cycle entirely and enter the G0 phase, and that is where it's no longer actively preparing to divide. Let's take a closer look at the first stage of interphase, G1. The G1 phase, also called the first gap phase, is the first stage of interphase. It will also typically be the longest phase of the cell cycle. For example, in humans, this will last about 8 to 10 hours, and during this time, the cell grows and prepares to replicate its DNA. The most important activity during G1 is cell growth. The cell will also synthesize many new proteins, especially enzymes that will be required for DNA replication. The cell also increases its ATP production or energy supply. This energy will be essential for the later stages of interphase. And before the cell can move on, it must pass through an important checkpoint. This checkpoint ensures the cell is healthy, large enough, and ready to begin DNA replication in S phase. The S phase, also called the synthesis phase, is the second stage of interphase. This is usually the second longest phase of the cell cycle. For example, in most human cells, S phase lasts about 6 to 8 hours. During this stage, the cell synthesizes or replicates its DNA, creating identical copies of each chromosome. The most important activity during S phase is DNA replication, also known as DNA synthesis. While we won't go into detail here, the DNA molecule will unwind and enzymes will help synthesize a new complementary strand. By the end of S phase, the cell contains two identical copies of its DNA, and this setup is what makes mitosis possible. In addition, during S phase, an organelle called the centrosome is duplicated. Here we have a graphic of what a centrosome will look like. And centrosomes help organize structures called spindle fibers, which will later play an important role in separating chromosomes during mitosis. We'll take a much closer look at how spindle fibers work in a future video. And lastly, before the cell leaves S phase, it checks for errors that may have occurred during DNA replication. This helps reduce mutations and remind us that the cell cycle is a very highly regulated process, not a random one. The G2 phase, or second gap phase, is the third stage of interphase and the final stage before mitosis begins. You can think of G2 as the cell's final preparation step. One of the main activities during G2 is continued cell growth. The cell increases in size and builds the materials it will need to divide. And as the last step before mitosis, one of its most important functions is that it will increase in size. Not only that, the cell will also synthesize proteins required for division, and organelles are duplicated so that each daughter cell will have the structure it needs to function. The cell will also check for and repairs any errors that may have occurred during S phase, especially errors in DNA replication. G2 contains another important checkpoint. At this checkpoint, the cell confirms that DNA replication is complete and that it is fully prepared to enter mitosis. Only cells that pass this checkpoint are allowed to continue, helping ensure the production of two identical daughter cells. The next and final stage of the cell cycle is M phase, or better known as mitosis. Mitosis is not a part of interphase. In this stage, the cell actively divides its genetic material in the cytoplasm into two identical daughter cells. Mitosis is divided into 5 subphases, including prophase, prometaphase, metaphase, anaphase, and telophase. In these steps, the cell will divide its genetic material inside the cell. After telophase, something called cytokinesis occurs, and this is where the cell divides its cytoplasm and is the final step that results in the actual two identical daughter cells. And this process looks different in different organisms, like in plants and animals. Mitosis is an important part of the cell cycle, and this is only a very brief overview of the steps that makes mitosis possible. In another video lesson, we will dive much deeper into what happens during this stage of the cell cycle. Let's take a look at this graphic again. The cell cycle starts at G1, where the cell grows and prepares for DNA replication. Then the cell will head over into S phase, where the cell replicates or synthesizes its DNA. Next, we'll head over into G2, where the cell will check for errors, grow, and prepare for mitosis. And once G2 has happened, the cell will enter mitosis, where it's actively divided into subphases. The end result of this will end up in two identical daughter cells. And once that has happened, the cell cycle will repeat. This helps organisms grow and repair. And every now and then, you will have a cell that leaves the cell cycle, ending in G0. I know that was a lot of material to cover, so let's check some of the important concepts. What is a somatic cell? A somatic cell is a body cell like your skin or liver. These are the cells where mitosis happens. What is the purpose of the cell cycle, and what is the result after one cycle? The purpose of the cell cycle is for organisms to grow and repair themselves through the cycle. It's how a human starting from a single cell can grow into you and me. The result after one cell cycle is two genetically identical daughter cells. And last question, how does the cell cycle help multicellular organisms with growth and repair? The cell cycle helps with growth because cells divide from one cell into two, then 2 into 4, and so on. Also, when cells get damaged or worn out, the cell cycle helps replace those old cells. Thank you so much for sticking around to the end, and I hope you found this video on the cell cycle helpful. If you haven't already, there are free downloadable resources including a vocabulary organizer and a cell cycle graphic organizer. There's also an optional paid downloadable guided notes that have all the main concepts from this video. Next video, we are going to explore more in detail some of the checkpoint mechanisms that cells use to ensure that cells reduce errors in this process. Thank you so much and keep learning. You're doing great. Bye!